Next paper, communication between the lower iliac tract and central nervous system, functional mapping and periaqueductal gray matter, presented by Meriaux and co-authors.
Thank you for the introduction. Good morning, everybody. So I'm Céline Meriaux, and I'm a neuroscientist. I'm working at the Department of Urology at Maastricht University, and I have nothing to disclose to declare. Well, when I started to work at the University of Maastricht, it was well accepted that an alteration in the bladder-brain axis would lead to some pathologies such as the overactive bladder syndrome complex. And this is why we decided to initiate a joint project with the Department of Translational Neuroscience at Maastricht University in order to study the communication between the lower urinary tract and the central nervous system. And we focus especially on the periaqueductal gray. So why the periaqueductal gray? As you can see on this framework, the periaqueductal gray play a key role in the spinal brainstem spinal system that control the micturition. So in one hand, the periaqueductal gray will receive the sensory information from the bladder, and on the other hand, the periaqueductal gray will communicate with the higher area of the brain. When the processing of all this information will be done, the periaqueductal gray will stimulate the pontine micturition center, and via the different nerve will control the micturition process. So when we look a bit closer to the periaqueductal gray, this structure is in the brainstem, and it's constituted of 4 different columnar subdivisions: the dorsomedial subdivision, the dorsolateral subdivision, the lateral PAG, and the ventrolateral PAG. And this subdivision has been established according to the nature of the type of neuron that are present in this subdivision. And all this subdivision has their own functions. So our question was which subdivision of the PAG and which type of neuron are involved in the processing of the afferent information from the bladder. For this, we opted for the following strategy. So we did electrical stimulation of the urinary bladder in order to induce some sensation in the bladder. And we combine this with topographical localization of CFOS in the PAG in the rat. So the CFOS is a well-known marker for the neuronal activity. So we had 32 animals, and they were randomly assigned in 5 experimental groups. The control group first did not undergo any surgical procedure. And for the 4 other groups, a bipolar stimulation electrode was implanted in the bladder wall. So there was 2 types of implantation. Or one of the electrodes was implanted in the dome of the bladder, and the other one was implanted in the base of the bladder. Or there was the 2 electrodes implanted close to the urethra, so at the base of the bladder. For 2 of the groups, there was no electrical stimulation, so these animals were called SHA group. And for the 2 other groups, they were stimulated, so electrical stimulation, and this group and these animals were called stimulated animals. So when we look at the experiment, first there was the implantation of the homemade electrode. So for this, the wires were attached to the urinary bladder wall, so as mentioned previously, and the wire were then tunneled in the back of the animal, and a plug was anchored to the skull of the animal in order to connect the plug to the stimulator and to do the stimulation in awake animal. Then, after a period of 2 weeks of recovery, stimulation was administered to the stimulated group during 1 hour with the following parameters. that we established to lead to a subtle contraction of the bladder without increasing the voiding frequency. And after 1 hour and a half, that is the optimal time in order to start the the activity of the neurons in the PAG. We sacrifice the animal by perfusion, and we process the brain for C4 staining. So we then took some picture of the sections, and what we found is a higher C4 positive expression within the periaqueductal gray when you compare the stimulated group with the sham group. And when we look at the caudal part, so at the posterior part of the PAG, we could find that in the ventrolateral PAG, there was a higher and a strong density of C4 positive cells close to the aqueduct, so in the medial part of the PAG. When you look at the statistical analysis, in the rostral PAG, in the anterior part of the PAG, for the animal that had implantation of the electrode in the dome and the base of the bladder, there was a higher number of C4 positive cells. It was statistically higher for the stimulated group compared to the sham group for both lateral PAG, dorsolateral PAG, and dorsomedial PAG. For the animal with the implantation of the electrode in the base of the bladder, in this case, there was a higher number of positive cells in the stimulated group compared to the sham group for the subdivision, so lateral subdivision and the dorsomedial subdivision. The statistical analysis in the caudal PAG revealed that for the animal with the implantation of the electrode in the base and the dome, there was a higher number and statistically higher number of C4 positive cells in the stimulated animal compared to the sham group for the ventrolateral PAG, lateral PAG, and dorsomedial PAG. However, for the animal with the implantation in the base of the bladder, there was no significant difference between the stimulated animals and the sham animals. So to summarize, the bladder contraction induced by electrical stimulation lead to an increase of the activity of the neuronal activity in the periaqueductal gray, especially in the caudal part of the periaqueductal gray, in the ventrolateral subdivision, and especially close to the medial part, so close to the aqueduct. We know from the literature that the ventrolateral subdivision of the PAG has different function and that different types of neurons are present in this structure. So what we would like to do now is to characterize these activated neurons, the one that has been shown to be activated after the bladder stimulation. And for this, we can combine the stimulation of the bladder with immunohistochemistry or in vivo electrophysiology. And would be interesting also to study the connection between the periaqueductal gray and the higher area of the brain. And this in order to be able to develop more specific options for the treatments of urinary dysfunction, such as pharmaceuticals or targeted neuromodulation. Thank you very much for your attention.
Thank you. Thank you. Open for a question.
Sakakibara from Japan, and congratulations for your very excellent papers. And just a comment, we previously, several years ago, we performed the experiment with cats of single neuronal firings recording within the PAG. And the results are almost the same with you. Just the ventrolateral and the caudal part, in the ventrolateral and the caudal part, there are many single neuronal activity in relation with the bladder storage and storage and micturition. So, thank you very much.
Question?
Hi, Thaddeus Spring from Medtronic. Very nice presentation. As you rightfully point out, periaqueductal gray has a lot of different functions that it's working with. So in fact, there's pain modulation as well as urology related as well. Given your stimulation amplitude, what was the behavioral reaction of these animals during the hour of stimulation? Because I think that starts to tell about what different functions you might expect to be related to the CFOS activation.
Well, in fact, we did some behavioral tests with the animals. So I just did the experiment just before coming to the Congress. So I have already a clue, well, just by observing the animals during the stimulation. And there was not, so this structure, so for example, the lateral PAG and the, well, in general, the dorsal part of the PAG, the dorsomedial or dorsolateral, are involved in the what they call fight-flight behavior. And in fact, we could not see anything of that. So yeah, as you can see in the results, there was an activation. It seems that there was an increase of activation in this region. However, with the behavioral test, we could not see any difference because we did the test before the surgery, after the surgery, and during the stimulation, and we could not see any difference in anxiety in these animals.
Thank you.
Karl-Erik Andersson, Aarhus. If I got it right, when you stimulated the dome, you get activity in the PAG, but if you stimulated lower down, you didn't. Is that correct?
Yes.
When you stimulated in the outflow region, for example, or the lower part of the bladder, you didn't get any reaction, force reaction. How could you then explain that all the sensory nerves in the bladder are in the lower third of the bladder? Is there a good explanation for this?
Well, thank you for a question. Well, I think, in fact, that it's due more to the field that we created. In fact, when you put the electrode in the dome and in the base, you have a really large part of the bladder that can contract. However, when we put the two electrodes close to the urethra, in this case, this is only a small part of the bladder that can contract between the two electrodes. So probably the contraction was not high enough in order to induce activity in the periaqueductal gray.
Yeah, but then you assume that all activity is related to the contractile activity. But if you stimulate electrically, you also stimulate the sensory nerve directly, and they are not related necessarily to the degree of contraction.
Yeah, well, in fact, what we wanted to do is to create a contraction that was not natural, like you can find in a normal bladder. So it was a way to induce a sensation without any pain, because sometimes some teams that use some stimulation that could be painful, and the problem is that the periaqueductal gray is involved in the processing of the pain. So this is why we decided to do this electrical stimulation in order to induce contraction. However, yeah, I think that this is how we can induce sensation, yeah.
Well, thank you very much. I think it was a very nice presentation.
Thank you.
Thank you very much. We need to move on. Thank you very much.